So today is my great pleasure to have both Barbara Stranger and Chris Katsapas speak. They're going to talk about the genetic regulation of gene expression, a very hot topic. Barbara is an assistant professor at the Brigham in the Division of Genetics and had a landmark paper in Nature that went over this at a genome-wide scale. And Chris has also worked on this to a great extent, particularly in type 2 diabetes, but in other fields as well. He has just taken a position at Yale, where he's an assistant professor, and we're very happy that he was able to return to the Broad 6 weeks after his move so that we can see him again and he can share his wisdom with us. So with that, I will have Barbara start, and it was just announced, this is all being recorded. If you need to have a question at the end, please step up to the microphone and ask the question. Barbara, thanks for coming.
Okay, thank you for that introduction. So today, Chris and I are going to be talking on the genetics of gene expression, a topic that a lot of people have been hearing about lately. We're really approaching this as a tutorial, so some of it will be pretty basic, but we'll try to touch on some of the key issues and how one might go about doing these kinds of projects and analyses. So the outline that we'll follow is what you see here. So we'll start discussing some of the basics of how you do these analyses, some of the evolutionary and population biology of genome regulation, how regulatory biology functions on a genome-wide scale, and also how these types of analyses and data can help inform GWAS results. So Chris and I are actually going to split this lecture in two. So I'll do the first half and he'll follow after. So I'll start discussing the basics right now. So first of all, we're really interested in the idea of quantitative traits. And quantitative traits are those that vary on a continuous basis, so not dichotomous, that can be measured quantitatively. And these kinds of traits are widely important across a variety of different aspects of biology, so agriculture, medicine, as well as ecology and evolution. And certainly you can imagine traits in agriculture that are important, like growth or yield, disease resistance, stress, so environmental stress, and in medicine, which we're most familiar with, disease susceptibility, drug response, or environmental response, including aspects of diet or exercise, might be quantitative traits. And they're really important in these studies of ecology and evolution because many of these types of traits are actually components of fitness, and so can influence how an organism or population or species evolves. So fundamental questions about quantitative traits is there a genetic basis to the underlying quantitative traits? And really, is there a way to identify this genetic basis? So how one goes about identifying a genetic basis is through the procedure of QTL mapping. And QTL mapping stands for quantitative trait locus mapping. And so what a quantitative trait locus is a region of the genome that harbors a variant that affects a quantitative phenotype. And so really in its simplest form, we have to identify a phenotype that you can measure numerically, and then you have to identify variation in that phenotype among individuals, variation in genotype among individuals, and then calculate a statistical link between the genotype and the phenotype of interest. So some of the questions that come out when we think about the genetic architecture of complex traits are those that you see up here. So really, how do variations in pathways or networks, and as well as interactions, contribute to a complex phenotype? Where are the variants that contribute to these phenotypes? Where are they located in the genome? Do they tend to be coding? Do they tend to be non-coding? And really, what types of variants affect the trait? Again, thinking about coding, not coding, conserved, not conserved, things like this. So this question of really where are variants found that underlie quantitative traits? Well, we know that there's approximately 25,000 genes in the human genome, and really that's a very, very small percentage of the entire genome. So most of the genome is not coding sequence that we assume is functional. And from conservation, so looking at genome sequence across multiple species, we actually see that non-coding regions of the genome, when you look across quite a long evolutionary distance, human, monkey, mouse, elephant, wallaby, you can see parts of the genome that are non-coding but that are highly conserved. And so these signatures of sequence conservation across such a wide phylogeny are indicative of function, we assume that they have remained stable over time because they have a function. And this gives us a clue that, in fact, there's important functional elements outside of coding regions. So why study gene expression? Well, we know that, in general, normal cell health and function depends on having the right genes turned on at the right time and in the right place. And we know where these patterns are perturbed. Sometimes there can be quite deleterious effects in humans seen as disease. And these don't have to be huge changes in gene expression levels. Even sometimes something that needs to be very tightly regulated, even very small changes, can have deleterious effects. Yet, despite this, we see that there's widespread variability among individuals with respect to gene expression levels, healthy, normal individuals. And this is seen in every species that's been looked at. There's a heritable genetic basis to this variation, and much of the variation, much of that genetic component has been able to be mapped. And where it's been able to be mapped, we find that much of it is located cis or nearby the locus in question. Now, we'll get back to this question of whether or not most regulatory variation is close to the gene or not when we go further through the talk. But we find that some of the variants that are associated with disease are also those that associate with gene expression variation. And this is something that Chris will get back to a little bit later on. So when we think about how genetic variation might contribute to gene expression variation, there really are several different ways. And this cartoon is really by no means comprehensive, but should just give you a flavor of how a nucleotide variant might have an influence on gene expression levels. So with this cartoon gene with a regulatory element upstream, you can imagine a mutation or a variant that would alter pre-mRNA splicing, or in the UTR, affecting localization of that mRNA. You can imagine a mutation that in the coding region, that if this is a protein, might change the conformation of that protein. If that protein were a transcription factor, it might bind differentially to another gene's regulatory element in a different way than if it were mutated or not. And of course, polymorphisms in the actual regulatory regions themselves might affect gene expression levels. Now, it's not only single nucleotide polymorphisms that might have effects on gene expression variation, copy number variation might. And this is a relatively newly appreciated form of common genetic polymorphism in the genome. And really, when people think about how it might affect gene expression, most people think of this very simple case of gene duplication. And if you have more copies of a gene, more expression. But really, all other kinds of copy number variation could have effect on gene expression just by changing the regulatory landscape of a gene and its regulatory elements. So an insertion might change the distance between regulatory elements in a gene, might add an extra exon, might delete a repressor. So these kinds of things show that there really are a lot of ways that genetic variation might contribute to gene expression variation. So how do we go about doing one of these studies? Well, first we have to identify or quantify mRNA levels. And so we consider the mRNA level as a quantitative trait that we're going to map. We survey that gene expression on a genome-wide scale across a set of individuals, some population size sample. And we can do this either using microarrays. That's traditionally how it's been done, but the newer technologies allow for RNA sequencing data. We survey genetic variation in the same individuals and map the responsible genetic factors. Okay, so what we're looking for is something in this very cartoon example right here. This would be a single SNP that would be leading to a difference in expression. And so individuals that had this would be an AG polymorphism here. Individuals with the G polymorph or GG homozygotes would have low expression. The heterozygote would have medium expression, and the AA homozygotes have high expression. But really, how we actually would do this is we use, you can use univariate or multivariate statistics. I'm really just going to show you the very simplistic form where we use regression or correlation-based methods. So this is essentially the same kind of slide we were just or same kind of scenario we were just looking at, where there's a population with and AG polymorphism and differences in mRNA levels. We would look in a population and take individuals who were homozygous for the A and plot their expression levels, take individuals heterozygous for the AG, plot their expression levels, and the other homozygote as well. And so what we get out of this, we can either do a linear regression or a Spearman rank correlation. You get p-values, you get a Spearman's rho, you have measures of the effect size, as well as how well your model explains the variation. So we do that on a SNP-by-SNP basis. So essentially, a whole genome eQTL analysis for a single gene is a genome-wide association study. And so what we would be doing is walking through the genome, SNP by SNP by SNP, asking whether the genetic variation at that location correlates with expression levels of the gene in question, okay? But when we do this, it's for many, many, many genes. But the way that we go about this with calculating the p-values and handling our data control, all of this is very similar to what needs to be done for a genome-wide association study. And Paul DeBacker's given some nice lectures here in the primer explaining some of the considerations to take into account, including the SNP quality control, definition of your phenotype, population stratification, and so on. So how do we interpret the p-values that we get out of these associations? And I'll get to that in a second when I talk about multiple testing. But the thing to keep in mind, again, is this is a genome-wide association study, and so if we do define a particular SNP as being significantly associated with gene expression, we have to keep in mind that this is really a marker. It is tagging a haplotype. It may be, if you are lucky enough, it may be the causal SNP, but and more likely it's not. Okay, so if we are doing, in practice, a whole genome eQTL study, we will have interrogated gene expression of all the genes in the genome and of a particular cell type or tissue type. So after quality control and determining which genes are even expressed in the sample, we go through and we do the genome-wide association study for each one of those traits. So it could be up to 15,000 GWAS essentially in a single experiment. So some terminology that you're likely to see and that I use as well, but that you'll see in the literature, is whether things are called a cis-eQTL or a trans-eQTL. And these are somewhat arbitrary distinctions because really every group defines them in their own way. But conceptually, the idea is that a cis-eQTL means that there is a variant located near to the position of the gene that has an effect on the gene expression. And there are specific types of variants that you might expect would be at those locations. So promoter polymorphisms or small insertion deletions, potentially epigenetic marks, altering epigenetic marks there. Trans-eQTLs now, on the other hand, are saying that the position of the functional variant is not near the gene locus. And so these could either be regulators or transcription factors, direct or indirect interactions. Okay. So really, when someone does a cis-eQTL analysis, the idea would be to test SNPs within a predefined distance of the gene. This is somewhat arbitrary. In my own research, we've defined a 1 megabase window upstream and downstream of a gene. And really, the idea there has been that we know some of the long-range enhancers in the human genome can act up to a 1 megabase distance. But really, every group decides this on their own. So when we do these kinds of analysis, you understand that there are a lot of tests that we're doing. If you do a genome-wide analysis, you're testing maybe up to 1 million SNPs, maybe with imputation now off 1,000 genomes data. You can have maybe up to 10 million SNPs. And so we really have to be very careful about multiple testing. And so we can apply different methodologies, including a simple Bonferroni correction, false discovery rate, or we can permute data. And we can either do this to the whole genome analysis or specifically a cis analysis. And the reason people would separate these two out is that by focusing in on a smaller region, the cis analysis, it's essentially like a candidate region study. And we know that there's a higher prior probability of being, or that functional elements are concentrated around genes. And so in a sense, we're sort of making a candidate region approach for each gene, but we have higher power because we're doing fewer tests. But really, any of these methods can be applied. The permutations on the whole genome level, though, is pretty infeasible still to do any large number of permutations for every gene, for every SNP. OK, so here's just an example, a real example of data. This is a gene that was surveyed in the Caucasian individuals of the International HapMap. And we had done a cis-eQTL analysis. And what you're looking at is genomic position on this axis. And each one of these dots represents a single SNP that we looked at. The red is meant to be where the gene itself is located. It's not to scale, clearly. And then on the y-axis here, we've got minus log 10 of the p-value. And so the higher up you go, the more significant the p-value is for each one of these SNPs in the region. And the line here is the permutation threshold that we had set for this particular gene, or that came out for this particular gene. OK. Sorry, I lost the screen here to see what I've got. OK. So, what you see is that there's a set of SNPs that all surpass our significance threshold. And what you're looking at in this plot here is the most significant SNP plotted for each individual that was interrogated. So this looks very much like what I showed in those cartoon examples. OK. So I'm going to shift gears for a second and move on to talk a little bit about the evolutionary and population biology of the regulatory variation. So that same gene that we were just looking at, that I said was in the Caucasian samples of the HapMap, was also looked at in 3 other populations of different ancestry. So in Han Chinese, in Japanese from Tokyo, and in Yoruba from Nigeria. And so the gene didn't show up on this plot for some reason, but is approximately right here. It was right near this big peak in the Ceph population. And so you can see that there is overlap across populations in the association. It's not showing up in the Han Chinese, although there seems to be a bit of a signal here. And so we can see that there's actually a lot of sharing of these associations. So there's common genetic variation across populations that is having effect on gene expression variation. And I wouldn't worry if anybody happened to notice that this seems to be going in a different direction than the other ones. It's just because the most significant SNP in the Yoruba was a slightly different SNP than the others. And just the way it's plotted shows it in a different direction. It's the same effect, though. OK. So now that the HapMap has moved on from those original 4 populations, we've now been able to do a cis-eQTL analysis across 8 different ethnicities. And I'm just going to walk you through what these abbreviations are. So these are the Caucasian individuals of European ancestry, Han Chinese from Beijing, Gujarati Indians in Houston, Japanese in Tokyo, Luhya in Kenya, Mexicans living in Los Angeles, Maasai in Kenya, and Yoruba in Nigeria. And so what you're looking at in this plot was 2 different permutation thresholds that we set that have different false discovery rates. You see the number of genes that had a cis-eQTL at the different permutation thresholds. So this is certainly the more liberal threshold. But what I want to draw your attention to are some of the numbers down here. So in this particular analysis, we tested approximately 15,000 genes that were expressed. These are lymphoblastoid cell lines. There are approximately 15,000 genes expressed from each of these populations. And so what we're seeing is that a huge proportion of them, maybe 70–80% of them, have a cis-eQTL across populations. And if you're happier with this kind of false discovery rate, something like 20%. We see quite a bit of sharing across populations. So there are associations that are shared in all 8 populations, really of different ancestry. But if we go to this level of shared in at least 2 populations, there's really quite a lot of common variation contributing to gene expression variation across populations. And so what we took out of this is really that this is only one cell type. Keep in mind, this is only one cell type. And only 8 populations. And the number of individuals varies from about 100 to 250 or so per population. That really there's a lot of diversity out there, functional diversity out there. We've only been able to look in this one particular cell type in these 8 populations, but really we're seeing that almost every gene will probably have some kind of cis-eQTL with common variation. Okay, so here's just one small example of a single cis-eQTL that was shared across all 8 populations. One of the questions that when we talk about cis-eQTLs or QTLs being shared across populations is the direction of the effect the same? Because that's really the hallmark of a replication. So what we're looking for is an association in population 1, also the same association detected in population 2, but the direction is exactly the same and not the opposite. So from those that I showed you before, if we plot all of the pairwise comparisons of populations and we say, we detected a cis-eQTL in 2 populations, was the direction the same? If it was the same, you would see dots in these 2 quadrants here. This is the Spearman's row. And so what you can instantly see from this plot is that almost all of the dots are in those quadrants. There are a few that are a few individual associations that are in opposite directions. It doesn't mean these are wrong. There are real biological ways that could come about, and differences in linkage disequilibrium between populations could cause a flip of direction. But what we're really seeing is the majority are shared in direction, so real replication. But one of the other things we wanted to ask is, okay, so the association is in both populations, but is the magnitude of the effect of that particular SNP, is it the same, or do different populations have modifiers that really change the effect slightly? And what we're plotting here is fold change between the 2 homozygote categories of a cis-eQTL that's shared between pairs of populations. And if the effect size is the same, you'd see it right along this diagonal. And so you can see from this plot that almost everything's right along the diagonal, but there are some interesting cases, like this one here, where the effect size is very different. The fold change is very different between 2 populations. So these are kind of interesting things as well. One of the things that comes out really of every eQTL study done so far is that the really big signals tend to be located at the transcription start site. So what we're looking at, if we look in one of these quadrants, is all of the significant genes that were detected in the Ceph population, and where the most significant SNP is located relative to the transcription start site. So this is the 0, so upstream, downstream from the transcription start site. And we're just showing the most significant SNP per gene and its level of significance. So really, you see this in every population. The big signals are right at the transcription start site, but in all populations, we have associations as far out as a megabase, which was the window that we tested. If we make the window larger and just keep expanding out, it does tail off. The density does tail off, but these are real effects that are significant really by permutations all the way out to a megabase. Okay, I'm not going to say too much about this right here, except that with such a large data set and from populations of diverse ancestry, you can start to say what kinds of genes tend to be differentially regulated between populations, because you can imagine that different populations have experienced different pressures and so on. So we see a lot of very general Go term categories related to gene expression variation, or related to gene expression, that tend to differ between populations. But we can also look at specific populations and see certain categories that stand out. There's certainly more to be done with that, but there's a lot of data there. Now, I just wanted to say a little bit about evolution. And Pardis Sabeti has been doing work looking at signals of natural selection in the human genome, and has developed methods for very tightly pinpointing regions of the genome that appear to have been under selection over time. And so here is a plot showing her score for selection. This is a CMS score, and so along a position on chromosome 12. And so what you see is there's a location right here that appears to have a very strong signal of selection. Well, we went, and she was interested in what could possibly be going on there. Well, she found that this particular signal happens to be in this gene PAR, and that this is a gene that has a role in T cell activation. Well, we tried to line up the eQTL data with the signals of selection data, and what you see is that this particular location is also an eQTL. And so it very well may be that there's a signal for selection that's related to gene expression and related to the immune function. So I'm going to wrap up this introductory part right now and just give you a couple take-home messages. So really, there are a lot of cis-eQTLs in the human genome. Chris is going to talk a little bit more about trans, but really with the little bit of sampling we've done, and there aren't that many different tissue data sets out there yet, we see that regulation by common variation is very common throughout the genome. And we see that a lot of the signal is really concentrated at the transcription start site, but there are long-distance acting regulators. We see that there's a lot of shared common variation across populations, functional variation, which isn't surprising given what we know about shared variation in general, but it's good to see that it's actually shared functional variation. It's quite interesting. And then finally, there are examples of regions of the genome that have been subjected to natural selection that appear to also be related to gene regulation, and so functional variation being selected on over time in the human lineage. So with that, I will move on and let Chris take over, and we'll answer questions at the end.
Good morning, everyone. So what I'm going to give you a flavor for is a couple of the problems that we can start talking about and posing questions in a more specific way with eQTLs. And the two things, can everyone hear me, by the way? No? Oh, cool. Does that? Yeah, it is on. It's just not close enough to me, I think. OK, so the two main motivating questions that I'll sort of take you through is, can we learn anything about how genes are regulated and about what the broader sense regulatory landscape of cells is from looking at eQTLs? And can we use eQTL data and expression data to sort of help us derive hypotheses about what biology might be being perturbed by variants that modulate risk to disease? So things that we map in association studies when we map higher level phenotypes like diseases, if you look at expression data, can you then sort of help craft biological hypotheses about what might be going on? So those are the two questions. And we'll start with regulatory biology. And I'll use primarily model organism examples to sort of help you think about this. This is applicable to humans, of course. We're not that different. But I think there's been a little bit more done right now in model organisms. So we'll talk about model organisms. So some of the early forays into this were done in yeast, actually crosses between lab and wild strains. This was done primarily in Leon Krugliak's lab, going back a fair number of years now. And the experiment is fairly basic. You take two completely different strains of yeast, you do a cross, you derive offspring that are a genetic mixture of the two very different parental strains, and you then genotype those and measure gene expression. You can do a mapping exercise in the same way as Barbara has told you. So for those of you who are a little more familiar with these designs, it's basically it's an experimental cross that you'd do in the lab. So very simple. We start with a fairly small sample. There's about 6,000 genes in the yeast genome. They mapped all of them. There's a whole bunch of them, like a lot of differential expression between the parents. You then say, OK, if they're different between the parents, is that difference genetic, and do they therefore map? And so quite a large number of them, considering we're only looking at 40 offspring, map. We get a lot of eQTLs for those SNPs, and about 30% of them have a cis signal. The rest appear to have trans, and there's a whole bunch of them that don't look as though they're different between the parents, that when you start mixing the backgrounds, do segregate, and they look different. So that's kind of interesting as well. This number is something I'll come back to a couple of times, there's an awful lot of interest in what the proportion of cis versus trans effects are. Most of the numbers are kind of in the less than 50% is cis, everything else is trans. That number varies widely depending on who you ask. The best calculations, the most sophisticated calculations have been done by Alkes Price and his collaborators so far in humans and to a lesser extent in mice, and these numbers are kind of right. So we see a fair amount of cis effects, which means that the genetic factors that are affecting gene expression map somewhere close to the gene, so it's either in the gene itself or somewhere immediately around. But there's a lot of trans variation as well, so points in other regions of the genome that are controlling gene expression. So if you then ask in the yeast data, if you then ask, okay, let's look at all of the trans signals anywhere and ask, is there buildup? So are there regions of the genome that seem to control the expression of lots of genes? So are there hotspots of trans activity? The answer is yes. So there's a whole bunch of groups in yeast. So there's 18, 20, sort of tens of genes that seem to be controlled from a single locus, which is kind of arguing that there is something at that locus that happens to be genetically variant that controls the expression of lots of genes. The easiest way to think about this is a transcription factor. And Barbara sort of gave you an example of that. If you can imagine that a transcription factor that has a coding change, which alters, let's say, its binding affinity to its regulatory elements, it then, when it's in play, it then affects the expression of all of the genes that it regulates. There are all sorts of more complicated models that you could postulate for that, but that's kind of the simplest way you can think about it. And if you look at the annotations of a lot of the genes that are influenced, they seem to share biological function in some way, and there's all sorts of cellular processes that happen. So this looks as though it's a viable mechanism, at least in yeast, for us to start teasing out what sets of genes are co-regulated, what biology they might be affecting, and sort of start putting together the regulatory architecture of cells. And if you look at it more pictorially, you can basically cluster. Each row here is a gene. Each column is an individual in which that gene's expression has been measured. And you can see there are low expressors and high expressors. I apologize to those of you who are red-green colorblind. But you can see you can sort of imagine from the different shades that they are grouped, all of these genes grouped together in low-expressing clusters and high-expressing clusters. And they actually map in aggregate, sort of this whole group will map to a region of the yeast genome, which also argues for this sort of large-scale regulatory variation. Now, if you look at mice, which is sort of more complex organisms, you kind of see the same story. So if you play the same game and ask, are there pileups somewhere in the genome of eQTLs so effects of controlled genes, you get these pileups in the mouse genome as well, and you get very large proportions of genes that apparently map here. So we're talking about effects that are 25% of genes seem to map to these regions. Right? We're talking about hundreds to thousands of genes in some instances. If you look at a different cross in mice done by a different group, you also seem to see these effects. And what I'm showing you here is this is the position of the genome at which a transcript maps. This is the position of the genome at which the transcript is located. And so if the effect is in the same region as the transcript itself, it'll lie on the diagonal. So those are cis effects. And if you get these big lines on the verticals, those argue for trans effects. And what you can see just sort of really clearly here is that there seem to be lots and lots of trans effects. And there are certain loci that control literally thousands of genes, which is potentially very interesting and highly unusual. Now, if you look at one of the more interesting mammals, which are humans, you don't see this. Now, you don't see any evidence of of regions controlling thousands of genes or hundreds of genes. There's the odd pileup of tens of transcripts, sort of like we saw in yeast maybe, but proportionally, there's nothing on this scale that we've seen in mice. Now, why could that be? I mean, it's clear that we're not that different from other mammals. So why can't we see these in humans? And there's several reasons, some technical, some probably biological. So one of the technical problems with a lot of these experiments has been that there are batch effects in the early data, especially in the mouse data. Sort of things were done in successive rounds of lab work, and it turns out that there are batch effects between those groups of data. The normalization and the pre-processing of data was fairly crude at the beginning of this decade, and so those signals were often not adequately accounted for. And so what that induces is a correlation structure between the first set of mice I did and the second set of mice I did. And you can imagine if the genotypes are slightly different between those two groups, it generates genetic signal. And one of the lessons we've learned the hard way is that these experiments need to be very, very delicately controlled in order to avoid very large-scale effects like that, which is something we didn't know before, because in gene expression effects, when you're not doing genetics, if you're just comparing case and control groups, this is not such a big problem. It turns out that in the genetic case, this is a problem. You will find a SNP that will describe your batch effect, basically, which means that you will map things. Replication, for these reasons, replication has been hard. A lot of these effects haven't been replicated, and so I think what it turns out is that the stunningly novel biology we thought we'd found in many cases is not there. The difficulty is that these trans effects tend to be weaker. And as far as we can tell, an individual effect on a gene expression level is going to be much weaker because it gets buffered by the system if it resides somewhere else in the genome than if it's cis, if it's proximal. And your biological intuition will tell you that if we're thinking about a transcription factor that's variant and therefore resets the level of a gene's expression level, well, a gene's expression level is not just controlled by one transcription factor. It's controlled by a whole array of factors, including 5 to 10 transcription factors, the transcriptional machinery itself, stochastic processes, and so on. And so you'd expect that to be a relatively weak effect on the overall expression level. If it's something in cis, so if we're talking about a UTR effect that just makes the transcript degrade much faster, you'd expect that to be a much larger effect. And so cis effects, biologically plausibly, will be larger than trans effects. Basically, what that means is it's harder to find trans effects. And so if it's harder to find trans effects, then of course it's going to be harder to find pileups of them. We're also looking at relatively small sample sizes in humans. In mice, it's relatively easy to generate 1,000 or a couple of thousand of individuals and do a mapping study. In humans, it's a little bit harder. This is being addressed. There's also, as Barbara mentioned, there's also the issue of tissue specificity. So gene expression is controlled differently in different cell types is the general idea here. And so that's something we need to think about. And you can see this from a lot of work that was done in Barbara's old group, where if you look now across 3 cell types in humans, and these are fibroblasts, lymphoblastoid cell lines, and freshly derived T cells, and you ask the question, OK, let's do eQTLs and ask how many are shared between all 3 tissues, any 2, or 1 tissue, you see that the vast majority of them are, in fact, tissue-specific. And that makes a lot of sense. You wouldn't expect the regulation of genes to be the same in T cells and neurons, for instance, or in T cells and B cells. They're different cells. They probably do different things with different genes, and quite often different things with the same genes. But that kind of clues us into what exactly regulatory biology might have in store. So we all kind of tell stories about how genes might be used in different contexts, in different cell types. And eQTL analysis actually opens up a window into that that's very powerful. So we can start looking at these sorts of differential regulation stories and actually derive a lot of the cell type specific biology that you might not have access to otherwise. So this is a very promising type of approach. Now, the other kind of set of questions you might ask is, can we use gene expression as one form of data to help us think about genome-wide association studies of larger the phenotypes? And so, again, you can play games like this, and sort of geeks like to play games like this, where you can describe all sorts of models. You can have, in each case, you've got something that's a SNP. This affects gene expression that winds up affecting a trait. So the trait is caused by an alteration in gene expression. You could have a SNP that causes a trait that then causes changes in gene expression. So all the shifts in gene expression are epiphenomena of the fact that you've changed the trait. You could imagine that if you're predisposed to an inflammation, then because you do tend to have inflammatory episodes, you'll have a whole host of different gene expression in your T cells, but that's just because you're prone to inflammation. It's not the different gene expression in T cells that's causing inflammation. It's the inflammation that causes the difference in gene expression. So gene expression becomes an epiphenomenon. What you can't do really is have a trait that influences SNP, and you can't have gene expression that influences SNP. So you've got a subset of possible hypotheses. You've got another set of hypotheses where you have a SNP that influences gene expression and influences a trait, but the two have nothing to do with each other. So they're two independent effects. And you could imagine more complicated sort of little models one could propose where you've got a SNP that influences a bunch of genes that cumulatively influence risk to a trait. You've got a bunch of SNPs that influence a gene that influence a trait, and all sorts of more complicated permutations of this thing. What it boils down to is that by using all these sort of slightly complex models, you can distinguish between them. So these are mathematical representations of these biological hypotheses, but you can actually distinguish between them given data. And so I can actually tell the difference, at least theoretically, between whether a SNP causes a gene expression change causes a trait, or a SNP causes a trait causes a gene expression change, because the data will be more parsimonious with one explanation than with the other. So here's a real-life example. So one of the early genome-wide association scans in Crohn's disease pinpointed a region that contained a gene and this region, there were variants. There was actually a SNP immediately upstream of the IRGM gene that predisposed to risk of Crohn's. And when people looked at this a little bit more carefully, they found that SNP actually was a marker for a very large deletion immediately upstream of the gene. And so the deletion basically takes out the promoter of this gene completely. And you can see that the gene's here. This is the size of the deletion. So we're talking about 20 kb worth of genome that's missing in some people and not missing in others. And whether it's missing or not seems to correlate with risk to Crohn's disease. So a very natural question you might ask is, OK, if we take out this 20 kb chunk immediately upstream of this gene, do we see a difference in gene expression? And does that have any evidence that that's what's driving the risk to disease? So the gene's called IRGM. The trait is Crohn's. And what you find is that the allele just quite literally takes out this 20 kb chunk, but it's marked virtually perfectly with a single SNP that has a lot of evidence of association with Crohn's disease. So if you go through and ask the question, OK, is the effect of is the expression of IRGM different conditional on the presence or absence of these 20 kb, then the answer is a resounding yes. And I won't take you through all of these data, but suffice it to say there's a huge difference in expression. OK, so we've got a region that associates to trait that also has an induction of gene expression. The really cool thing is that when you go in and knock down this gene in a cell system, you find a biological difference. So this is no longer genetic. We're not looking at this SNP, so we can't really, the SNP is no longer a factor. You're going in and knocking down the expression of this gene. The risk allele is a low expressor, and if you decrease the expression of this gene in a cell system, you find that there's a biological effect. And the biological effect is bacterial autophagy. So in cells of the gut epithelium, one of the things that you see is that there's a very specific type of endocytosis of bacteria called autophagy, where bacteria try and invade cells. As they're invaded, the membrane pinches off, and the cell, as a defensive mechanism, creates a second membrane around the invading bacterium. This causes an envelope, which is then trafficked to a specific kind of lysosome for destruction. And this is basically how these cells deal with a bacterial assault. You'll remember that in the gut, there's a lot of bacteria, and cells just routinely do this as bacteria try and invade, whether they're pathogenic or commensal. They just get trafficked and destroyed, and cells kind of survive. And what you find is that if you knock this gene down, this process is affected, and it's defective. And so Crohn's is an inflammatory disease of the gut, and specifically the gut lining. And it's very attractive to sort of think that, you know what, if you take this gene out, or you decrease the expression of this gene with a genetic factor, then you have a defect in this process. A cell is unable to clear the normal bacterial load that it gets. And if that's the case, then perhaps what you set up is a chronic inflammatory phenomenon. And that might help explain why some people are more susceptible to an inflammatory bowel disease than others. That part hasn't been quite proven yet. That's speculation, but it certainly makes sense from a biological hypothesis. And so this is an indication where an eQTL has direct link not only to a biological phenomenon, but also to risk of disease. And so you will imagine that there will be at least some other examples where we'll be able to help elucidate what the genetic causes of disease are, or disease risk are. This won't work for all of them. I'm sure that there will be plenty of examples of genome-wide association signals that are not mediated through gene expression, but some, probably hefty proportion of them will, and we can learn a lot of biology by integrating these data. I'll just leave you with the idea that this is going to be important to look at specific cell types for this. Because of the tissue specificity of gene expression variation that I showed you earlier, you could imagine that if you look in the wrong tissue, you won't be able to see the signal. Basically, the experiment that you'd want to do to correlate trait mapping to expression mapping is a comparison. So if you have your expression genome-wide association zoomed in on a locus, so you've got one track that is for the expression trait, you've got another track that is for the disease trait, and basically what you'd be looking for is correlation between these two expression values. If you look in the wrong tissue, of course, because eQTLs are tissue-specific, the expression signals are going to be flat, because there's no genetic effect in that tissue. And so the tissue specificity is going to be important. That's going to make it challenging to look at certain diseases. The psychiatric diseases spring to mind immediately, where brain tissue is going to be a lot harder to come by. I think some of the iPS stem cell models that are being created now will probably help us a lot in this thing, but in that respect. But this is going to be a bit of a bugbear for us. Surmountable, but something that we're going to need to think about. And so I will just leave you with the slides for various interactive resources that will give you, in a web interface, user-friendly sort of way,